Hi everyone, this is Dr. Jyoti. I welcome you all on my YouTube channel. In this session, we are discussing about the 10 most common type of PCR technique which are used in biotech lab and biotech industry. So let's start the session. As you know, PCR is a very important technique which we utilize in biotech sector. So if you are in a biotech lab or in the biotech laboratory in the R&D sector, you need to amplify DNA for your various projects, and for that purpose we need polymerase chain reaction. So here we are exploring 10 of the essential types of PCR which are quite common in the biotech lab. The number one is the standard and the conventional PCR. This is mainly consist of three main steps, that is denaturation, annealing and extension. In such kind of PCR which is routinely utilized in the lab, you perform 30 to 35 cycles of PCR amplifications. The second most common PCR is your RTPCR, which is also called as reverse transcriptase PCR. Mostly RTPCR is used to detect the RNA expression level and also the viral load. So here you have to first convert your RNA to cDNA and then amplify the target sequence. So in most of my aptamer project, we have utilized the traditional PCR also along with the reverse transcriptase. The another significant PCR is your quantitative PCR, which is also called as qPCR. qPCR measures the amount of DNA in the samples using either fluorescent dye and other alternative dyes. The another PCR which is significant one and advanced one is your digital PCR, which is also called as dPCR. As I said, dPCR is an advanced form of PCR. It offers precision, sensitivity and also reproducibility in comparison to your traditional PCR method. It also can detect the rare mutation, copy numbers variation and also quantify nucleic acid with greater accuracy. Now moving ahead, the next PCR is your nested PCR. Nested PCR amplifies specific DNA fragment and also increase the sensitivity and specificity and minimize non-specific amplification. Basically, they are used in research also and in for diagnostic purpose. The another one is your multiplex PCR. Multiplex PCR enables simultaneous amplification of multiple targets in one reaction and thereby improving the efficacy and also help us in conserving crucial samples. The next one is your inverse PCR. Inverse PCR is a technique used to amplify DNA when the flanking sequence is already known. This kind of PCR is used for cloning and studying the chromosomal rearrangement. And the next one is your long-range PCR. Long-range PCR is used to amplify fragments up to 40 kb in the length, and it requires special enzymes and buffer system for optimal performance and standardization. I hope you have liked the session and you are going to explore them further. If you have any queries related to these techniques, you can contact us. Thank you so much.